All right, welcome everybody. Can you guys hear me? All right, excellent, thank you. All right, welcome everybody to the 5th Protein Engineering and Design webinar organized by the journal Protein Engineering, Design and Selection, or PEDS for short. My name is Roberto Chica. I'm the editor-in-chief of the journal, and it's a pleasure to see so many of you here today. So before we get started, I'll just like to say a few words about the journal. So as you may know, this journal was founded in 1986 by and for research scientists. And since then, it's established itself as an excellent destination for articles that relate to the study of protein engineering and design. So basically understanding the fundamental link between protein structure, function, dynamics, and evolution, as well as papers describing the engineering of novel proteins with interesting properties. Now, since I took over editorship of this journal last year, I've implemented a few changes to make this journal even more exciting as a publication destination for our community. And I'll just briefly state 3 of them. The first one is we've streamlined the review process to make it more efficient. That means that it's faster than it used to be. The average time to first decision is now a little below 6 weeks. And we ensure that you get expert feedback that is fair. Secondly, we've moved to an online-only publication model, which means that your paper gets put online as soon as possible so that everybody can read it quickly. And third, we've eliminated all publication charges except for the optional open access fee. So we hope that with those initiatives, it gives you even more reason to publish with us. And there's a few more things I'd like to mention. The first one is that we have a special issue on what I call hot topics in protein engineering and design. These are topical reviews on different topics related to protein engineering and design. And these should be coming out in the next few weeks, the first batch. And they will be made freely accessible by everybody. So I think there's going to be a little bit of everything for everybody in there. And also, I'll remind you that we have a Twitter account. So if you search for PEDS, Protein Engineering and Design, you'll find us. And on Twitter, that's where we announce all of our activities and initiatives, including the next webinars, the papers that are coming out, the editor's choice articles that we make freely accessible to everybody. The special issue will be announced there, etc. So please follow us if you haven't already done so. Okay, so now I'm going to give you a little bit of instructions on how the webinar will go. So first of all, we're going to have a short presentation, about 20 minutes, from the first author of the latest Editor's Choice article that was published recently in PEDS. This will be followed by a short question period. After that, we'll have our keynote presentation by Dr. Andreas Plückthun. This presentation will be about 40 minutes, followed again by a 5 to 10 minute question period. And as typical, will be done in about an hour and a half total for the whole webinar. So if you have questions, I encourage you to use the Ask a Question button that's at the bottom of your screen and type your question there. You can do this at any moment. The presenter will answer the question at the end of the presentation. You can also use the chat box on the right. But ideally, if you ask it in the Ask a Question button, it will make sure that we get to those first. So I encourage you to use that instead of chat box. But if for whatever reason you use the chat box, we can also see that. Okay, so that's how it's going to go. So I'm going to, I guess, introduce now our first speaker, Dr. Alex Sevi. He's a research scientist and protein engineer at Singular Genomics in San Diego. And Alex earned his undergraduate degree in biochemistry and molecular biology at the University of Georgia. From there, he moved to a PhD at Vanderbilt University, working with James Crow and Jens Myler. And during his PhD, he developed computational methods for the design of multi-specificity in antibodies. So a lot of nice work there. If you're interested, you should look up those papers. Then he moved for a postdoctoral fellowship at Merck, again, working on the engineering of antibodies for binding specific molecules and still using a computational and an experimental approach. And since 2020, he's at Singular Genomics as a research scientist, and he'll tell us today about his editor's choice article that was published, I think, a few weeks ago on PEDS. And this article is freely accessible if you want to read it after the presentation. All right, so take it away, Alex.
All right, thank you, Roberto, for the introduction, and thank you for selecting the article as the editor's choice and also for the invitation to present here. It's a really great honor, so I really appreciate it. And thank you to all the organizers for putting together this webinar. So like Dr. Chica mentioned, I'm going to present some of the work I did at Merck, working on the structure and sequence-based design of synthetic antibody libraries. So I'm going to be talking today about a really interesting molecule called a VHH antibody. So these are single domain antibodies that consist only of a single heavy chain. And so if you look at the schematic here on the left, you have a normal human IgG. So this is a pair of heavy and light chains, total about 150 kilodaltons. And so the interesting thing is that different species like camels, llamas, and alpacas naturally make heavy chain only antibodies. So they make these llama IgGs that are just two heavy chains paired together. These are much smaller, only 80 kilodaltons. And from these, you can actually derive only a single domain, which is just the binding portion, which we call the VHH. So this molecule is about 15 kilodaltons, and it has very similar binding characteristics to a full human or llama IgG. So you can make these against any targets that you want, with the advantage that you don't have to worry about heavy and light chain pairing, and you have a single well-folded molecule that can accomplish the same binding characteristics. And these molecules are nice because they're very stable. They naturally have pretty high homology to human antibodies. So for potential uses of therapeutic, this is very helpful. They've also been really useful in crystallography. So as small reagents to help bind possibly flexible proteins to help them form crystals more easily, or to help in determining the structure, these VHH are really useful. And they've been around for quite a long time, been validated against a lot of different targets in an academic setting. And just recently, over the past couple of years, they've started to emerge also as therapeutics that have gone through clinical trials. And I believe the first therapeutic VHH was recently approved. So these molecules are really starting to make it to the big stage now. Traditionally, the way for identifying VHH is through immunization. And so in this kind of scheme, you take a llama or an alpaca that you immunize with the antigen of interest, collect the PBMCs, extract the mRNA, clone these genes into a display vector, either yeast or phage, and then go through subsequent rounds of panning to identify those that bind to your antigen of interest. And this works really well. You end up with really high affinity hits out of this process, but it has some limitations. So as you can imagine, the process is pretty time consuming and expensive because you have to maintain a large animal facility with all of these llamas and alpacas. There's no guarantee of what you're going to get out of the end. So you may have some really highly mutated VHH that wouldn't be suited for therapeutics for immunogenicity purposes. And it may be limited to certain antigens. So certain antigens may not be stable circulating within an animal, so they may not mount a sufficient immune response. So for these reasons, even though the immunization-based approaches have been really successful in the past, there is a need for a different approach to try to get to the same end. So at Merck, we were interested in developing a synthetic antibody library approach to developing VHH. So the way that this approach would work is that we build a synthetic antibody library. So these are genes encoding antibodies built completely from scratch that we clone into a yeast display vector. So then these molecules are displayed on the surface of the yeast, and we can build libraries up to about 10 to the 9th, 10 to the 10th in size. The yeast cells are displaying different antibody variants. We can then bind those to an antigen labeled with biotin. And once the cells are all coated with antigen, at least those that are specific for the antigen, we can use streptavidin conjugated to a fluorophore and use flow cytometry to sort out the cells that are expressing variants that are specific for our antigen. And we can go through subsequent cycles of enrichment to end up with a highly enriched pool of antibodies that are specific only for our target of interest. And so overall, the goal of this project was to develop a synthetic VHH library for expression on the yeast cell surface to identify high affinity binders. And overall, I was going to take a two-pronged approach. So I was going to use structure and sequence data. The big challenge here is to decide how do we build these libraries in a way where they're going to be highly productive and there's going to be sufficient diversity in the antibodies so they can bind a wide variety of targets, but not too much diversity so that there's not productivity in the library. And so the challenge is really to introduce diversity only at the positions where it's most likely to improve antigen binding. And in this study, we're using a synthetic library licensed from Harvard Medical School from the Andrew Kruse lab. This is kind of the standard in the field. There's a lot of great papers coming from this lab and using these libraries. So we're going to use the Andrew Kruse library as a kind of gold standard to compare our libraries against to see how well do they perform. And so as I mentioned, I'm going to use a two-pronged approach for trying to design the library. Overall, I'm going to organize this talk. The first half, I'm going to describe the design of the libraries and how we design them. And then the second half, I'm going to talk about the experimental work where we actually built the libraries and screen them against a few test campaigns. So the way I approached this problem was to first look at the structure of known VHH. So if we look at the PDB and pull out all the VHH antigen structures that are in there, we can use computational modeling using Rosetta to analyze these complexes and try to design where the optimized diversity can be placed. So what amino acids are most likely to contribute to antigen binding. I'll go into a little bit more detail on that. And then also use the sequence-based approach from NGS repertoires of alpacas and camels to see what do their CDR loops naturally look like. So the first question that I wanted to ask when starting this project is which region of the VHH is responsible for binding? So there's a lot of data about human antibodies, but since VHH are just a single heavy chain, it's likely that they're going to engage their targets in a totally different way than a human antibody would. So to get at this question, I first took all of the VHH antigen complexes, so these were 208 different complexes, and modeled these using Rosetta, and then calculated what's the predicted binding energy of each residue all along the VHH, and then grouped those by region. And so here on this bar chart, on the x-axis, I'm showing the different regions of the VHH, going from framework 1, CDR1, framework 2, CDR2, and so on. And on the y-axis, I'm showing the average contribution to binding energy on a percentage basis. So the way to interpret this chart would be CDRH3, on average, contributes about 60% of the binding energy to the complex. So a few take-home messages from this analysis. Number one, not surprisingly, but the CDRH3 dominates the interaction with the antigen. So like I mentioned, about 60% of the interaction energy on average is coming from the CDRH3. And this is pretty much in line with what's out there in the literature, what's known about VHH. Another take-home message was that on average, the CDRH1 and 2 contribute about the same amount of interaction energy on average. So one question was whether we could prioritize CDR1 over CDR2 or vice versa in building the libraries. And from this analysis, it seems like we couldn't prioritize one over the other, that in general, they tend to contribute about the same to the interaction. So after doing this, I wanted to get a little bit more granular. And so now instead of looking at overall by region, I actually broke down by individual residue. And so here on these charts, I'm showing across the x-axis is the position along either the CDRH1 or the CDRH2. And on the y-axis, instead of percentage binding, I'm actually showing the energy contribution to binding. And since these are energy measures, lower energy means stronger binding. So the residues with the lowest energy on average are the ones that tend to contribute most strongly to binding. And so a couple takeaway messages from this plot. Number one is that not all of the residues contribute equally. So certain residues such as number 26 on the CDRH1, 51 and 57 on the CDRH2, on average tend to contribute very little to the binding energy. So these residues are more involved in the stability of the molecule itself, usually aren't involved in antigen binding. So this is good to know. We're probably not going to build diversity into these positions. But there are some residues that tend to contribute more strongly. So I've highlighted here 4 residues that on average contributed the most energy to the binding interaction. And so from this analysis, these 4 residues would be the candidates for introducing diversity into the libraries. So now that I've done the structural analysis, I moved on to a sequence-based analysis. So this is a analysis of VHH sequences from 2 public databases to look at what are the different characteristics of these repertoires. And so I've cited here the 2 papers that the sequencing datasets come from. Thank you to the authors of those papers for making that data available. So one of these datasets is from a naive alpaca, and the second is from naive camels. So from analyzing these sequence datasets, the first question is, what does the IGHV and J gene usage look like in alpaca? So here I've shown a heat map where on the y-axis is shown the different V genes, the x-axis is shown the different J genes, and from about 10,000 different sequences, we can see where do most of the sequences get their V and J genes from. So this is pretty interesting because if you look at the similar heat map from a human naive repertoire, it's going to be much more diffuse. So you're going to have usage of a much higher number of V genes and J genes. It's going to be a lot more spread out. So it's a little bit surprising to look at the usage in alpaca that it's very highly restricted. So there's really only one square here that has a high percentage of usage, and that's the V3S53 V gene and J4 gene. So the overwhelming majority of the sequences are using this particular combination of V and J genes. So this was really interesting and led us to believe that these genes are used so highly in a naive alpaca. These are probably good germlines to use the basis of a naive library. The next question is, which positions in the CDR1 and 2 can we actually randomize without compromising the stability of the molecule? So here I'm showing a sequence logo where on the x-axis is different positions across the sequence, either CDR H1 or 2. And on the y-axis, I'm showing the bit score. So this is a representation of how frequent each amino acid was seen in the sequencing data set. So the height of each letter corresponds to how often we observe that amino acid. And a few observations from this data. Number one is that a few amino acids seem to be pretty well conserved. So G26 is very rarely mutated. Similarly, I51, G55, and T57. Those are pretty much conserved more often than other residues. So we can kind of infer from this that these residues are going to be really important for maintaining stability of the molecule, and these would not be good positions to put in diversity. And if I overlay the same 4 bars as candidates from diversity from the structural analysis, it turns out that these 4 positions are really highly mutable also on a sequence basis from this data set. So this would indicate that since these are mutated so often, that they're probably going to be mutated, easily mutated in response to an antigen. And so this really goes well in hand with what I observed from the structural analysis, in that these 4 might be good candidates to put in diversity into the libraries. So the last element that I wanted to introduce is humanization of the VHH frameworks. So this is important because when you're building a therapeutic, you always want to keep in mind what the end molecule would look like. And generally, you wouldn't want to introduce a fully alpaca molecule into a human as a therapeutic, because it may induce an unwanted immune response. So to get at this, we were thinking, well, what if we just humanize the sequence beforehand, and then anything that comes out of the library is already going to be humanized, and we wouldn't have to humanize it further. So to get at this, I took the same 3S53 gene, aligned it to the closest human homologue, and I show that alignment here. This is human gene 323, allele 4. And in vertical bars, I show all the differences between the alpaca amino acid and the human amino acid. And from the literature, it's known that you need, typically there's 4 amino acids from the VHH that are considered essential. So you need these 4 amino acids to be the alpaca identity for the molecule to be able to fold by itself. These are amino acids that are right at the heavy chain light chain interface. So if these are humanized, they probably won't fold into a soluble molecule. So every amino acid other than these 4, and then also built one with only 2 that we hypothesized could still be soluble with only 2 alpaca amino acids, were reverted to the human identity. So what we end up here is a framework that is almost completely human with the exception of either 4 or 2 amino acids. And this humanization was built into the synthetic libraries as well. So based on all of this analysis, here's the final library designs. And so these are differentiated by the amount of diversity. So either, I name these either low diversity or high diversity. The low diversity libraries are only using those candidates from the structural and sequence analysis, whereas the high diversity is kind of randomizing the whole loops. And then these differ by their use of framework. So either alpaca framework or humanized with 4 amino acids, humanized with 2 amino acids. And you can see that N transform library size for all these libraries was about 1 × 10⁹. So this is a good size. These are large libraries for a yeast display vector. And as I mentioned, as a comparison point, we're going to use the Andrew Cruz library, which also has about 1 × 10⁹ library size. So we're kind of, all the libraries are transformed to a similar size, and we can see what the end results look like. So before going through with the campaigns, we just did a quick NGS analysis of the naive libraries. So after transformation, we got between about 20,000 to 50,000 filtered high quality reads. And out of these, almost 100, very close to 100% were unique CDR3s. So 99.9, 99.8% were unique. So this is good because there was no error introduced when we're building the library where certain genes are favored over others. And the percent productivity ranged between 65 to 84%. So some of the genes had frame shifts or stop codons introduced, but the majority were still productive. So moving on, I'm going to quickly talk about 3 benchmark campaigns that I conducted to try to validate the library performance. So we picked 3 kind of model antigens to see what kind of antibody, what kind of VHH can we pull out of the libraries. And these 3 campaigns are roughly ordered from easy, medium to hard. So I would consider mouse PD1, very well characterized target. This would be kind of an easy campaign since it's just a soluble protein. A beta peptide, this is a short 40 amino acid peptide. So this is kind of a medium difficulty target. And then MRGX1 is a GPCR. And so as a membrane protein, it's a lot more difficult. So this I considered a hard target. So we have these 3 different types of proteins. Want to see how do the libraries perform against all 3. And here's how the campaign workflow went. So the first step was 2 steps of magnetic cell sorting to try to isolate those on a bulk basis to try to get the library size down. Next, we did 4 rounds of flow sorting to try to enrich the pool for only highly specific, high affinity binders. And we did single cell FACS, so instead of bulk FACS sorting, actually analyzing each individual clone to make sure that it is specific for what we think it is. And then the last step is recombinant expression in mammalian cell culture and characterization. So here, just briefly to introduce what the flow cytometry plots look like. So if we have a yeast cell displaying a VHH fused to an anchor protein, the VHH are expressed with an anti-HA tag. So this can be detected by, well, they're expressed with an HA tag, and they can be detected by an anti-HA tag. This is always shown on the y-axis. The y-axis is showing how many cells are actually expressing antibody. And then streptavidin, which is binding to a bipenylated antigen, is shown on the x-axis. So binding to antigen is always going to be shown on the x-axis. And therefore, the binders that we're really interested in are going to be up here in quadrant 2. So after 4 rounds of sorting for a campaign against mouse PD1, here are the final results of the FACS plots from this campaign. So again, y-axis is showing VHH expression, x-axis is showing antigen binding. Going across the columns, I'm showing the 4 different libraries that were designed for this study, and then the Andrew Cruz library as a comparison. The top row is showing dissorting conditions with no antigen present. So this is a really important control because we're just as likely to isolate antibodies that bind to streptavidin or bind to our fluorophore as we are to isolate antibodies that bind to the antigen itself. So we always have to do a no antigen control to make sure that we're not generating what's called reagent binders that are just sticking to some either streptavidin or fluorophore. And then on the bottom, these are really the plots of interest. These are the flow sorting in the presence of 50 nanomolar mouse PD1. And so this kind of quadrant 2 tail is the part that we're interested in. And so a few take-home messages from these plots. Number one is that from all 5 libraries, we're getting specific binding, so very little reagent binding, and lots of binders to mouse PD-1 specifically. It's interesting that our hypothesis was that the low diversity libraries would perform just as well as the high diversity, because we're putting the diversity only in the most critical positions from the previous analysis. And so this data would tend to indicate that that was successful, because the low diversity libraries tended to work just as well as the high diversity. So this would indicate to us that we're really putting diversity in the most critical positions. And another interesting observation was that the alpaca and the humanized libraries tended to perform just as well. So when we first started, had a little bit of concern that the humanization might compromise the stability of the molecule or cause libraries to be less productive. And from this analysis, it seems like the humanized libraries, either 4 or 2 amino acids, perform just as well as the alpaca. So this is really nice, because then we could, in future campaigns, only focus on the humanized libraries, which are really the ones that we're most interested in. So here is the characterization of these clones on a recombinant basis. So taking the clones from the previous flow plot, we expressed 37 of these as recombinant VHH and measured the affinity by Forte Bio. And so at x-axis, I'm showing different libraries, and y-axis is the affinity. So most of the libraries gave hits around 100 nanomolar affinity, which since we did all the sorting conditions at 100 nanomolar, the final sort was at 50 nanomolar. It's not surprising that this is the range that we got. And it seems very similar between libraries. There's not a significant difference between one or the other. So in the interest of time, I'm going to move kind of quickly to the next campaign. So these are the results from the A-beta campaign. And so this one is a little different, because we're sorting now against the peptide antigen, but we're still getting a really highly enriched pool of peptide-specific VHH. So there's very little background binding for the most part in the internal libraries. There's a little bit of background binding for the entry-cruise library, but still much more antigen-specific. And you can notice that previously we were using 50 nanomolar of PD-1. Here I'm using 10 nanomolar of A-beta peptide. So these are actually higher affinity against the peptide than they were against PD-1. So these are really, really high affinity binders that are coming out of this campaign. And the same recombinant analysis from the A-beta campaign. So here I made 42 recombinant VHH produced in mammalian cell culture, measured the affinity by Forte Bio. And here similarly, most of the final hits were around 100 nanomolar affinity, but we did get some that were down to double-digit nanomolar affinity, and got one that had single digits. So one of the libraries produced one VHH with an affinity of 5 nanomolar. So this was really good from a naive campaign for VHH. We were really happy about this result. And then the final campaign, just to touch on briefly. So this was the MRGX1 GPCR. So this was a little bit of a different challenge. Here we were looking for VHH to specifically stabilize the antagonist-bound conformation. This was a collaboration with the structural biology group in West Point, Pennsylvania. And so we did all of the antigen binding here in the presence of a detergent. Since the MRGX1 is stabilized in detergent micelles, we had to include detergent in every step. And also in the presence of the small molecule antagonist to try to get conformation-specific VHH. And here there's a little bit more background binding and then no antigen controls. But in the presence of our GPCR antigen, we're getting a really highly enriched pool of high affinity binders. And unfortunately, I couldn't include the recombinant binding data from this campaign. But similarly, we got some really nice high affinity binders to MRGX1 out of this campaign. So just some conclusions: use structure and sequence analysis to get the properties of naturally occurring VHH, built 4 synthetic libraries, verified their quality, and out of 3 discovery campaigns, we're able to get some really nice high affinity binders. And if you want any more details on this project, you can, I included here the citation for the paper that you can go back and read to get all the details. So with that, just want to acknowledge everybody at Biologics Discovery in Boston, specifically Ning Tang Chen, who is my postdoc mentor for this project. Really provided a lot of nice guidance and helped a lot with this project. So I want to give special thanks to him. Also to Michelle Tyler, Adam, Renee, Ginny, and Lei, who are all members of the team who contributed in different ways. And thanks to Harini and Andre from West Point, Pennsylvania. So these were from the structural biology group who helped with the MRGX1 campaign. And also thanks to CM Shea and Gary Starling, who are part of the executive team who sponsored the postdoc program, and overall to the Merck postdoc program. And so with that, thank you for your attention. Thank you for calling in, and I can take any questions.
All right, thank you, Alex. All right, I can see there are already a few questions for you in the Ask a Question button. So if you could read them out loud and answer them, that'd be appreciated. Thank you.
Okay, first question. Are camels the only animals expressing VHH, and do we know why they do? In addition, how long is the lifetime of this molecule in the human system? That's a good question. So the only animals that I'm aware of are the camels, alpacas, and llamas. There are different animals that express different forms of single domain antibodies that aren't exactly VHH. So like sharks express, I think they're called VNAR molecules that are single domain, but they're folded kind of in a different way. So different species have different antibodies that are folded in different ways. Do we know why they do? That's kind of an evolutionary question. I'm not exactly sure the answer to that, but that would be interesting of why that happens. How long is the lifetime of this molecule? So this is a big issue with VHH, and one of the big caveats is that they do have a shorter half-life because they lack an FC domain to do FC recycling. So there are a couple ways to get around that. You can either make these molecules as FC fusions that'll increase the half-life. People have made them multi-specifics fused to like human serum albumin, also increases the half-life. So that is a limitation, but there are ways around that. Next question, maybe I'm missing something here, but why focus exclusively on the design of CDR1 and 2 if CDR3 is the one contributing the most to binding and affinity? That is a great point. I kind of breezed over the CDR3 design, but the CDR3 was basically fully randomized. We included lengths from, I think, 6 amino acids up to 18, and those were fully randomized amino acids. So the CDR3s were basically completely degenerate, and those were the same between every library. So yeah, the CDR3 is definitely the most critical portion, but those were just fully randomized. How does the long CDR loop bind a peptide-like antibody? Is there any structural insight? That is a good question. Yeah, normally peptides bind kind of in between the heavy and light chain of a human antibody. So that's why I was a little bit wary starting a VHH campaign to see how well it was going to bind. Unfortunately, we couldn't get any structures for this project. That would be really interesting, especially to take some of the higher affinity ones and see exactly how does the peptide interact. I think I've seen some where it sits in a groove between the CDRH1 and CDRH3. So that could be one mechanism, but yeah, we'd love to get some structural data on those. One question, anything special on the high affinity binders to A beta? Do they recognize unfolded or amyloid structures? So we purposely designed the peptide in this campaign to be non-aggregated A beta. And then we did have some data on plaque staining for amyloid plaques. And some of the clones did recognize the amyloid plaques. I couldn't include the data here, but we did have some recognizing those, but those weren't included in the full campaign. And then some questions from the chat. Can you raise nanobodies against glycopeptides and in particular the sugar moiety? I definitely think so. I think traditionally, my impression is that glycopeptides are kind of difficult to do in immunization, but I think in a synthetic campaign, it's definitely doable. Basically anything that you can label with a biotin molecule, you can use in a synthetic campaign. And if you do your screening the right way, you could really make sure that they only bind to the sugar and not to any other part of the glycopeptide. So I definitely think that could be a potential application for this technology. And the final question, do you think the Rosetta positional analysis at the beginning shows large enough RAU value difference? Yeah, that's an interesting point. So the differences were kind of slight between the positions, and part of that is because we were averaging out. So looking at 208 structures and then averaging them, there's some that really heavily involve certain amino acids, some that really heavily involved others. So there was a little bit of leeway in choosing which amino acids were quote unquote the most interesting based on the structure analysis. So some of the amino acids could have been included because the RU difference was not huge, but we were just kind of looking for a kind of guiding factor to pick the most important ones.
Okay, great. Thank you, Alex. We're going to stop here in the interest of time. I noticed there is one more question that popped in the Ask a Question. So if you wouldn't mind during the next presentation to answer it by typing your answer right there. All right. Okay. Join me in thanking Dr. Sevi for his nice presentation. Okay, great. So now it's my pleasure to introduce our keynote speaker for today, Professor Andreas Plückthun from the University of Zurich. I mean, many of you know him. He's a pioneer in protein engineering. So I think many of you have read his work over the years. Nevertheless, I'll give a short biographical introduction. So Dr. Plückthun is from Germany. He studied in chemistry at the University of Heidelberg for his undergraduate degree. Then he moved to the University of California, San Diego for his PhD with Edward Dennis. After that, he moved to the East Coast at Harvard for a postdoctoral fellowship with Jeremy Knowles, who's a very well-known and respected enzymologist. And in 1985, he started his independent career at the Max Planck Institute of Biochemistry, and then moved in 1993 at the University of Zurich. And he's, as I said, very well known. He's a pioneer in the field, won many awards. I'm not going to mention them, but I'll just mention three important contributions, I think, to the field. Number one, pioneering work in the development of synthetic antibodies and using E. coli as an engineering platform. So that's very important work. Also, something that I know very well, given that my PhD advisor contributed to this, was the development of ribosome display, which is a selection method for enzymes. And lastly, what I think we may hear about today, which is the conception and development of DARPin, so Designed Ankyrin Repeat Proteins, which are antibody mimetics. And these proteins are really interesting because they can be made to bind other molecules with high affinity. So I'll stop it there. I'll welcome you again, Dr. Plückthun, and we look forward to your presentation.
Many thanks for having me. Thank you very much for this very kind introduction. And I will try now to share the screen. Let's see whether that will work. And theoretically, you should see this now. So today I would like to talk to you about a project which has been going on in our lab for a long time, which has to do with generating particular scaffolds and to address the problem of how to bind linear epitopes. So as we have already heard just now, the great number of reagents that are used in for lab work, for Western blots, ELISAs, and things like that are still created by immunization. And today, of course, we have already seen, and there are many other examples out in the literature, libraries have entered the scene, typically by the means of a universal library. And in these cases, sorry, this is the wrong choice. I wanted to put the laser up like that. From these universal libraries using selection technologies, we can also generate binders. Now, this is still the state of the art for folded proteins. And I think a lot of the therapeutics have actually come out this way, less so for lab reagents or for reagents used in experimental research. Now, there's a limitation here, and that is for each target, an individual immunization and characterization has to be done. And actually, this is also true for starting from a universal library. So that's not a problem if you have like 4 or 5 targets, but it is a problem if you're even conceiving of creating binders to, for example, a whole proteome or even a subclass of that. And that's essentially the question I want to address today. And so if we are looking now at binding to folded epitopes, we can use, for example, Designed Ankyrin Repeat Proteins, which you see coming around on the left. And you can appreciate that by using these fully synthetic systems, we can create binding surfaces which are extremely complementary to the target. They're not only complementary in the surface structure, but also in the surface composition, and really place hydrophobics and hydrogen bond donors and acceptors exactly where needed. So I consider this actually a solved problem. We have ways of creating libraries, we have ways of doing selection, and so on and so forth. So the typical way how to do this is to create a synthetic library, randomized from the structure where one thinks one should have that, putting the residue types in there that are compatible with this structure, and then use any one of these selection technologies. We particularly like ribosome display. This is something that we've developed, but sometimes we've also used phage display or yeast display for selecting DARPin libraries. And so this is still the way to do this for folded proteins. Now, that's the same structure, and this is a schematic of showing the same thing, which is that we have a folded epitope, meaning that the residues being recognized are only coming together in the folded structure, and we can also call this a discontinuous epitope. And so, like I already said, every target is a new project. Now, if we consider linear epitopes and we don't do anything else, this would be the same situation. We would still have essentially a new selection for every new peptide or for every new linear epitope. And the reason for this is that if we consider, for example, antibodies as they bind linear epitopes or peptides, that each peptide in each antibody structure has a different conformation. So it's a completely new molecular system, and we can draw no conclusions from having done this before. And so the question is, could we have an opportunity here? In other words, could we exploit the periodicity of peptides? Now, of course, you can't use antibodies for this, and you can't even use DARPins for this. But if we could do this, we could perhaps avoid selections altogether and use an engineering approach from the beginning to the end. And so the question is, what do we use instead? And clearly, we would have to come up with a binding scaffold that reciprocates the periodicity of the peptides, that is also modular and has the same periodicity as the peptide. Now you might say, well, this is of no interest because the important ones, the important epitopes are all conformational, and that's probably actually true for therapy. But there's a lot of applications where linear epitopes are very important. For example, if you think about detection and quantification of proteins, for example, by Western blots or other methods, they all require linear epitopes. Proteins there are denatured. Sometimes natural unstructured tails can be used, or even tags can be added. We all use this in the lab every day. Also very important, the epitopes for phosphorylation events are mostly in unstructured regions, typically in tails, in loops, in linkers, and so on and so forth. And very important, the epitopes for the epigenetic markers are also mostly in unstructured regions, i.e. the histone tails. So there's a lot of biological importance in detecting sequence-specific or modification-specific unstructured regions. And let me illustrate this with this cartoon. When a GPCR binds an agonist, subsequently the tail gets phosphorylated. That leads to the recruitment of arrestin. And actually what then happens downstream of this depends on what exact pattern of phosphorylation is put on. Not all the residues are used, only a subset is used. And so this creates like a barcode, and the cellular events that follow from this depends on what exactly has happened. And of course, that depends on the exact nature of the agonist. And so therefore, it would, of course, be immensely useful to be able to figure this out very fast and for a lot of receptors, and to really have a sequence-specific, very fast and very sensitive detection of such kind of events. And so this brings us back to the question, what scaffold should we use? And we turned to nature and noted that the armadillo repeat proteins are a very, very interesting starting point. And actually, the name doesn't come from the animal armadillo, but it comes from the fact that one member of that family is beta-catenin, and when this gene is knocked out from Drosophila, the larvae don't look like the wild type here, but look like this. And Eric Wieschaus, who discovered that, thought that this looks like an armadillo, albeit a very small one. And if we now look at the binding mechanism on these armadillo repeat proteins, we see something very fascinating. So this is a drawing from the structure, just simplified. And what we see is the peptide is actually stretched out pretty much. You can see here the backbone in purple color. And what we can also see is that every of these helices that make contact carry one asparagine that makes a double hydrogen bond to every second peptide group. So this essentially straps the peptide down on the armadillo protein. And that is completely conserved. Now, the side chains of the bound peptide, which you can see here in green, go alternatingly up and down. And they are actually contacting the yellow residues, and the yellow residues are part of the armadillo protein, and it's the yellow residues which actually give the specificity for what green residues are being recognized. And what you can already appreciate here is there's a periodicity there, and approximately 2 amino acids of the peptide are bound per repeat. This becomes more apparent if we convert this to a cartoon. And we see the peptide running from left to right. Again, in purple, you see the backbone. In orange, you see this conserved asparagine that makes this double hydrogen bond. In green, you see the side chains of the bound peptide. In yellow, you see the side chains of the armadillo protein, and that together is the complex. And here we can perhaps more clearly see the illustration of the repetitiveness. So the problem is, of course, that natural armadillo repeat proteins are totally irregular, like natural proteins are. And so in the beginning of this project, the goal was to convert these totally irregular natural proteins to very regularly designed proteins. And I spare you this work, which has been published in the past, and simply say that we've been able to do this and fast forward to what has become possible since. And one of the issues, of course, was what would these what these consensus proteins actually bind. And so we looked into the binding of the natural proteins, and especially the family of the importins that give rise to one of the major families. And if we look in very closely, they actually have, as I said, they're quite more irregular, have bound patches of lysine arginine. And we thought we'll perhaps make then a peptide that consists of lysine and arginine, and perhaps that is the desired peptide for the consensus protein. So I fast forward to the crystal structure of exactly that, which is the complex of a full consensus armadillo repeat protein and the electron density of the bound peptide, which now consists of alternating lysine and arginines. And you can appreciate that this binds in a very regular way. And if we go more closely, we can actually see that this is not just a non-specific complex of some sort, but really forms very, very defined interactions where, for example, this arginine side chain sticks in a pocket and sees this glutamate here at the depth, far away from the plane, and is flanked by these two tryptophans, creating this pocket. And here you can see the main chain, and here you can again see this double hydrogen bond that straps the main chain down close to the protein. Now, very interesting was the observation that there is a very regular binding. And what we have plotted here is affinity as 1/kd, so higher is better. Here we have the peptide length, and here we have the protein length. And so what you can appreciate is that with a given protein making the peptide longer, there is an increase in binding energy. That is not particularly surprising because obviously you have more interactions. What is perhaps not so immediately apparent is that it also goes the other way, meaning that you have the same peptide here and make the protein longer. You would think, well, you have actually the same number of interactions. Why should the binding get better? But you have to consider that, first of all, statistics favor the longer ones. We also have long-range electrostatic contribution. Is more is that the conserved asparagine has been mutated to an alanine, that we always lose exactly the same amount. So it makes a very regular contribution to binding. So it's a very regular system. Moreover, if we now use single residue mutagenesis of the peptide, we can also see a very regular binding. In other words, this is an alanine scanning of particular residue of this bound consensus peptide. And if we remove one lysine to an alanine in either position, we lose exactly the same amount of binding energy. If we mutate an arginine to an alanine, we lose a slightly larger amount of binding energy, but pretty much independent of the position. If we mutate two lysines to alanines, we can see that we lose exactly twice the amount of binding energy. If you remove twice the arginine, you also get the addition of the loss of binding energy. And if you mutate one lysine, one arginine, again, independent of where they are, you get also a very close to additive behavior. So we can see this is a very regular contribution from occupying each pocket. And we thought this is very exciting. Moreover, this together means that we can more or less choose the range of affinities where we want to be, where we want to do our experiments, and where we want to do our applications at. So if we make the protein longer and make the peptide longer, we have the option of using the exact same system and go anywhere from 5 micromolar in affinity to 5 picomolar. And we can, for every application and for every engineering experiment, choose the sweet spot of length where we want to do this. And if we're not quite happy, we can introduce an alanine, for example, to make the binding slightly weaker. So one of the interesting observations is the kinetics of this somehow weird, is it unusual? And so we decided to measure this together with my colleague Ben Schuler using FRET. And so the peptide is fused to GFP. The protein is fused to an acceptor dye. And when they come in contact, there's a FRET signal, so you can actually measure the increase at 594. And we can measure the dissociation by adding now an unlabeled excess peptide. And of course, then the FRET signal goes down again. And if we do this, we first of all observe that with this charge system, we have extremely fast on rates, very fast, I have to say. Several times 10 to the 8. We have decent off rates. And if we then calculate the KD, these are very interesting numbers. So we get down to the 10 to the minus 13, which I think is actually not so bad for a completely man-made engineered system. I think it actually compares quite well with what nature has given us. Now, there's another important thing which we need to understand before going on, which is there more than one orientation? So you might think that if we have this repetitive peptide, if we have this repetitive protein, they could possibly bind in different orientations. And so we thought we need to figure this out, and again used FRET for this, and again with my colleague Ben Schuler, but this time using single molecules for that. And so the donor-only signal would emerge at 509, the FRET signal at 594. And so if we have a movement and not in perfect register, we would expect that the donor-only signal is higher and the FRET signal is lower. And so we can actually see what kind of arrangements we have in this population. And so the question, is there more than one orientation? The answer is yes. We can see this. So we have really probably 3 orientations there. The majority appears to be the one that has the highest FRET signal, which is sort of the one in register. But we also have some that are shifted to the left and to the right, if you will. And so that would be a problem for the future engineer. So we had to solve that, and I'll explain in a second why and how. But let me just say that we have solved it. And so by having orthogonal binding pockets, you stop that movement, and thereby we call this a lock. And that prevents the sliding of the peptide. And you can see this now here. And now essentially all of the population is in the right register. So this is all good. So now the question is, how did we do this? And we again, first of all, turn to nature, looking at other classes of armadillo repeat proteins. In this case, the beta-catenins, which have a very different preference for what kind of residues they bind. You can see this is much more hydrophobic. There's an aromat and so on and so forth. And again, without going through all the details, what we did is we just grafted that on the beta-catenin type of on the importin type of consensus, and this site then was expected to recognize leucine, serine, phenylalanine. And in the context of an added octapeptide, we would expect an affinity, or we measured an affinity of 77 nanomolar. We determined the crystal structure. It pretty much looked exactly like expected from the graft. And then we thought, well, can we make this site even better and perhaps give it even more affinity? And then we turned to Rosetta Design and simply asked, well, can we improve the residues from this graft to perhaps something that is more commensurate with our peptide? And we did this over 4 stages, going from 83 to 56 nanomolar to 28 to 19. So that's already an improvement. So we went from this log now from 83 to 19. And then we thought, okay, now we have a slightly different binding site. Perhaps we can find an even better peptide. And so we did. And the peptide isoleucine threonine tryptophan brings this down even further. The amusing thing that we discovered then is that this particular binder actually cares about whether this one residue in the peptide is isoleucine or leucine. And that discrimination is actually even more than I had ever hoped for, that the protein can actually even discriminate between these 2 amino acids in the peptide, which are obviously very, very similar. We determined the crystal structure of that, and this is now the overlay with the design of the improved version. And if we do the crystal structure with the isoleucine threonine tryptophan version, we can see that this is again fitting very well with the expectations. And this immediately shows that the increased binding energy comes from the increased interactions that each one of these amino acids has in the now slightly improved pockets. Okay, so to summarize where we are now, we have stopped the sliding. We can keep them in one position and have them now in a way that they don't move back and forth, and can now address the question, can we change the specificity? But before this, we have to worry about another problem, and that is the distance of the protein has to fit the distance of the peptide very exactly. In other words, these asparaginins that we have said need to bind with their double hydrogen bonds these peptide bonds, and we cannot really change the distance between the peptide bonds. That's a given. And so the question is, are our asparagines exactly as the distance that they should be? And so obviously, if there were not, there's not that much we could do. We couldn't make the helix thicker. We cannot change the length of the peptide bond. And the only thing that we can do is change the relative orientation of the helices, which will, of course, change the relative distance of the asparagines ever so slightly. And so we were interested in just looking at some of the crystal structures that we had obtained in the meantime. How good or how bad are we? And the good thing is the protein does actually match the peptide repeat length quite well. And the peptide actually helps that once the peptide is bound, it actually adjusts the protein very close to where it needs to be. And so this is a great reassurance. And this very slight adaptability of the protein is a great advantage. And so this then was borne out by the crystal structures. And if we superimpose and morph the structure with and without bound peptide, that's the same protein with and without bound peptide, and just in a morph, we can see there's an ever so slight movement. And that's, of course, a morph of a crystal structure. So you might ask, well, is this also happening in solution? And so you can think about this like a leaf spring of an ever so slight adjustment if needed. And we were just sort of asking, is this also happening in solution? And again, got together with my colleague Ben Schuler, this time doing the following experiment, putting fluorophores on either end of the armadillo protein. and asking the question, if you add the peptide and there's an ever so slight adjustment, would you actually see this in solution? And the answer is yes. The FRET efficiency increases slightly from 0.21 to 0.31. And if we were asking what would we expect from the crystal structure, it would be very similar. We would actually expect 0.23 from 0.28, and within the experimental error, this is, I would say, in agreement, and says this very slight adjustment actually happens in solution as well. And so that's good. So in other words, there is some leniency within on our side. And of course, the RMSD of these proteins is only 1.5 angstroms. So in a typical protein design consideration, one would say these are the same, but there's an ever so slight difference. Good. So now we can really address the question of creation of new pockets. And that is, of course, has to be done, this is very important, in the context always of a whole protein. So we will redesign one pocket at a time, but always keep this in the context of a whole protein. And so one of the approaches that we've looked at is to say, well, we want to change the specificity for this amino acid here in red, and thereby randomize the yellow ones that are contacting it and are around it, and recreate a library. We also use yeast surface display. Actually, we used all methods that I've mentioned in the beginning. Yeast surface display, for reasons that I'm happy to discuss, actually has turned out best for this purpose. And so this is I guess, a very traditional setup where the protein, the armadillo protein, is displayed on the yeast surface. We have tags making sure that the thing is full length. We have the peptide labeled, but we're doing this whole thing in a very unusual way. So we have tailor-made libraries that only allow certain residue types, that we avoid stop codons. We have the prevention of sliding that I mentioned through the log sequence. We have a lot of effort in counter selection for specificity, because we're not really looking for the highest affinity, we're looking for the highest selectivity, which is a completely different problem. We need to have strictly monovalent targets, and we need strong fluorophores, because we cannot really use any amplification system. So there's a lot of technology development behind that, and I think we have figured it out now and have gotten a long way with that. And so let me show you some snapshots on the selectivity test of some clones. And so the first thing that in a typical work of this type is there would be a specificity screen on yeast that we just first of all see what's happening there. And we are querying peptides that differ exactly by one amino acid. So it's a peptide and everything else is the same except the one amino acid. And so, for example, this is a selection against valine, and this is just a clone taken from the yeast selection. And this particular clone prefers valine, as it should, and it also has some interest in threonine, which I think is quite acceptable because that's a very similar amino acid. And remember, everything else is the same. It's just this one OH group which differs, and that is a little bit worse, but it's I guess otherwise a very similar system. And so these are the different residues that are probed at this same position. This is one for tyrosine, which clearly prefers tyrosine only. And here are some other snapshots that give you sort of a flavor what can happen. So for example, this guy is a little bit less worried about aromatics and has also some residual recognition of valine. So not every clone right out of the bat is specific, but it has to really go through these additional counter selections that I have hinted at. Now, if we then go further and take these proteins out and express them in E. coli and purify them and characterize them directly, And again, using peptides which only differ in one amino acid. So for example, here we see that this one is totally selective for tyrosine. Again, this is 1/kd, so higher is better. And you can see this pocket really recognizes only tyrosine. It's not interested in anything else. And the amusing thing is it came out of a completely redesigned arginine pocket, but it has completely lost any interest in binding arginine. And you can see here the KD values in the context of this peptide. We then determine the crystal structure, and we can, I guess, understand why it has this specificity. It really likes to cradle the tyrosine quite well. It makes a hydrogen bond, and it's sort of very apparent why other amino acids wouldn't fit or wouldn't make any suitable interactions. And this is sort of a closer view of what's happening there. So we're really cradling this and making the kind of desired interactions with the OH group of that side chain. And that gives an explanation why it really cares about all details of that particular side chain. This is another example now going for tryptophan, which again comes out as the best one in a direct comparison. Here are some other ones. This is an isoleucine selection, which again has been, I would say, preferred isoleucine very much. It still has a little bit of a binding to other aliphatic amino acids, which I guess is okay. A valine selection as an example, which has some cross-reactivity of tyrosine. So we have to sort of characterize what's happening there. And that's sort of a selection which is less specific, but could potentially be interesting in other contexts. So this tolerates H-bond donors. So what I've shown you here is the redesign in a variety of selections of this pocket, which used to bind arginine, which we have been looking at in the last few slides. But there's another one going down, which typically binds lysine in this fully in this full consensus protein. And the problem with that is this is relatively shallow. So in other words, how to solve that? We actually created loops that wrap around the side chains and move over it to really have additional interactions from other sides. And again, there's been a lot of selections going on. And I'll show you an example of glutamine now, where this lysine is now a glutamine. And 3 different proteins that have come out of the selection, which are very, very selective for glutamine, which only tolerate glutamine here. So if you replace it by alanine, glutamate, leucine, or asparagine, you lose a lot of binding energy. And the reason why this is probably so pronounced is because this loop actually then grabs it from both sides. And you can see these are 3 different pockets that have different sequences and different loops and have all solved the problem. And so now the next challenge is, of course, to assemble them into a longer protein. And so I'm showing you the example now of assembling the binder of tyrosine. So this is the tyrosine pocket. And in order to create a double pocket, we want to have something where a tyrosine pocket, 2 tyrosine pockets are next to each other on this face here. And so the problem then, of course, is that you have to redesign the ridge between these 2 pockets. So this is a pocket, that's a pocket, and the ridge needs to somehow be a compromise between this and that. And so the ridge are these pink residues. And again, optimizing those, it does make a difference, but a solution can be found. And thereby we can create proteins that have now alternating residues with which bind several of these tyrosines. And one of the very interesting features is that this makes, again, a very, very high affinity system. So this is a system now where where 4 tyrosines are bound with a protein containing 4 tyrosine pockets. This creates an affinity of 9 nanomolar. Actually, the corresponding length of the purely lysine arginine system has 250 nanomolar, so this clearly binds better. And if we increase this to 6, we come to femtomolar binding and actually can no longer measure any dissociation. So this is essentially, for all practical purposes, under these conditions, an irreversible system, which is actually also very exciting. So we see no dissociation at all over 16 hours. And so obviously this is still part of an ongoing journey. So we create pockets now that I think we have figured out how to do this, specificity selections. We're still improving the combinatorial assembly strategies, but I think we have the foot in the door. Extracting information from next generation sequencing about correlations, anti-correlations, and very exciting also determinants which appear to influence affinity versus specificity, which is not necessarily the same. Trying to improve the speed of structure determinations, and of course collaborating with computational approaches. And perhaps this particular presentation will also encourage other people from the audience to become interested in this. So the outlook is, of course, going on, and the long-term vision here is to do all of what I just said only once. And essentially, in the context of longer proteins all the time, have now units that we can assemble for many dipeptides that we can put together in full proteins and do this, as I said, only once, and then basically assemble this off the shelf to have a given set of binding proteins that are put together from binding pockets to have then predictable binding of linear epitopes without doing any panning at all. And so I don't think we're naive. We completely understand that there are cases where this is totally modular, but there are also cases where this deviates, that 2 pockets can talk to each other. And I think we have some ways to deal with that. And it's, of course, also possible that the pockets talk to each other like that and to create double modules. And so really doing these kind of selections as well. And so I think the very long-term vision, and this is more for the reagent antibody field, perhaps than for therapeutics, would be that we get away from what's done today, that we no longer need to immunize hundreds of thousands of mice. And we perhaps also don't need to have doing panning from universal libraries one after the other, but really move gradually towards a technology where there's no target preparation, there's no selection, there's no screening. You only would need to know the sequence. In other words, the next coronavirus comes, it's sufficient that you know the sequence, and having these things tailor-made would be able to say one could create a binding protein off the bat in extremely short time. So that's the long-term vision. And of course, these are steps along the way, but I think hopefully one can create the impression that one is learning a lot of proteins, most importantly, how little we actually know. And so I think the most important slide is this one. I have the privilege of having a phenomenal group working with me, and the key people who did the last bit of this long-term armadillo journey, Yvonne Stark, who really figured out this selection strategy, Erich Michel, who figured out the loop stuff. I mentioned the work of all of these people flowing into the work. These are the people who have done the crystallography, I like to say that we actually have collaborations with previous speakers from this series, Birte Höcker, and some very early work with David Baker, Cyril Fleischmann, and Fabio Parmigiani. Actually, when he was still in my group, and with my colleague Amadeo Kaflisch, great library design from the group of Anna Hein, NMR collaboration with my colleague Oliver Zerbe. And so it's a great pleasure to work with these guys. I like to take the opportunity to thank them at this opportunity, and like to thank all of you for listening to me. And now we have to see whether we can get out of this. Stop sharing.
Great.
Okay.
All right, thank you very much for the excellent presentation. So we'll open up the floor for questions. I think there's already a few for you in the Ask a Question box.
Okay. So I guess I'll start at the top. I don't know. These are the still the questions for Alex.
Yeah, you may want to scroll down.
Maybe start at the bottom. Yeah, can you comment on whether or when flexibility either in the armadillo protein or the binding target may enhance binding? Yeah, I think that's a very interesting question, and of course the fair answer is quantitatively we don't know. And of course, in general, the textbook view is if you freeze binding, you lose entropy, and that's not good. On the other hand, of course, if you sort of are too rigid and you cannot adapt, you don't have any binding. And so, I mean, I think the flexibility of the protein is very minor. The flexibility of the peptide is enormous. And so I think it's very difficult to put quantitative terms on that. And I'm not sure whether I can really do justice to that question. Okay, so just on your penultimate slide and your comment, I'm not quite sure what the question is. Why high Why high selectivity? What's the advantage? So that's an easy one. So obviously, let's think about applications, and let's think about, let's say, Western blots. And let's say this we had more pockets already and could simply say we can design at least one binder for every human protein. And we could simply say, look, we have to be able to have this selectivity around the sequence to be sure that it is this isozyme and not that isozyme. And so I think that is the reason why, of course, highly selective antibodies, for example, in proteomics and any kind of protein analysis are extremely valuable. Okay, well, there's a lot of questions coming up. Yeah, so how do we imagine that this is working with peptides with strong secondary and tertiary quaternary structure? I mean, I'm not trying to see this in competition to the selection against folded proteins, because that we already have. We have selection systems, be it now from antibodies, from single domain antibodies, from DARPins, all kinds of things. I'm really talking about the situation where we have unstructured regions in, for example, for example, in the ones that getting phosphorylated, in the ones that have tails or are tags, and so on and so forth. And of course, if in a situation of, for example, a Western blot or something like that, of course the protein is denatured, and you already put in energy with the interaction with the detergent to do that. And so, I mean, I think the idea would not be to use the binding energy to unfold the protein, but really to use this in the situation of having an accessible loop, an accessible tail, or a strongly denatured form where the recognition of the sequence is the key. Aha, yeah. Do you think we can? Do I think that I can make binding pocket for every amino acid, including? Now it scrolled away. I guess including the glycines, prolines, and so on and so forth. Normally I would say no, but if we completely close the pocket, it's not impossible that we can have at least a strong punishment for anything greater than glycine, maybe alanine. And so, I mean, of course, we haven't done that. We so far have concentrated on the ones that have a have a notable side chain and haven't actually concentrated on the the very small ones, but we do have, of course, selections against serine, threonine, and so on and so forth. Can you comment if the armadillo scaffold has outperformed the MHC molecules for binding linear epitopes? I think that's a difficult comparison. Of course, the MHC molecules have a different challenge in the sense that they have a much higher binding energy. In other words, there's a much slower on and off rate. And so therefore, I don't think they could be. We have considered them in the beginning to use as a binding scaffold, but I think they were very hard to engineer for their makeup. If there is a strong barrier in binding and dissociation, I think that the system lives from being able to equilibrate. I'm not sure whether that sort of answers the question. Okay, I was wondering how parallel is the peptide along the superhelical axis of the repeat fiber, and how long can the repeat become before running into folding problems? Natural repeats, natural armadillo repeat proteins are very long. I mean, they can have 12, 14 repeats. These things exist. And having said this, of course, the natural proteins are the natural proteins are irregular. They always have some deviations from the sort of consensus scaffold. I have to remember how long the longest were that we have made. I think we have 2 capping repeats, and I think up to 7 repeats we have made, and they work fine. They can be expressed in E. coli, and so on and so forth.
And maybe I can sneak in a final question.
Sure.
So have you started also engineering these modules to bind post-translationally modified amino acids?
Yeah, that's one of the big goals. I mean, very clearly, what we want to achieve is to, of course, having like for example, the phosphorylated residues, and then I think the ideal thing would be to have the phosphorylated residues and the non-phosphorylated residues flanked by exactly the same residues to basically be able to do a ratio.
Mm-hmm.
Now we're working very hard on that. I'm not quite there yet. But I mean, this is one of the very clear applications that would, of course, be, I think, of great utility.
Mm-hmm. Do you think you'd be able to discriminate between mono, di, and trimethylated lysine?
I have to admit that we have started on that. So at some point, we may know the answer.
Okay. Excellent. All right. In the interest of time, unfortunately, we have to stop here because it is 90 minutes in. But I wanted to thank the speakers, and please join me in thanking them both for their excellent presentations. And I wanted to thank everybody in the audience for participating and asking excellent questions. And I remind you that there will be additional webinars throughout the winter and spring, all the way until April. So you can check the schedule on the PEDS website or the PEDS Twitter account. So thank you, everybody, and we'll see you next time.
Thank you.
Thank you.